So welcome everybody. I'm delighted to be here this morning to update you all with some new news from Biotron, which was announced to the ASX this morning. My name is Michelle Miller and I'm the CEO and Managing Director of Biotron. And joining me as well on the panel is Professor Rob Murphy from Northwestern University in Chicago. Rob is chair of our scientific advisory board and has an extensive background and knowledge of development of antivirals, in particular the HIV space, but also more recently with COVID. And we're delighted that he can join us this morning as well.
Good morning.
Thank you. So what I propose to do is go through very briefly some slides. I won't go through all of them. The main thing is they will then be in the public domain and people can, they'll be available on our website and people can look at them at their leisure. But what we want to tell people is the latest news that we've put out this morning about BIT225, which is our lead drug for development of HIV that's been in the clinic, been in 9 clinical trials, over 200 patients, has now shown very good results against SARS-CoV-2 in a COVID-19 model of animal model of disease. For those who aren't familiar with Biotron, we're a small molecule drug development company. We're not a vaccine company. We develop normal small molecule drugs that you take in capsule form as you would any other medication. We have a unique approach to tackling viruses. We combine attacking the virus directly to knock down the virus, but also then boosting the body's immune system to further knock down virus levels and boost the body's immune system to fight the infection. We're different from the other companies that are out there by targeting these proteins called Viruporins. They're found on a broad range of different viruses, and they're central to stopping cells that are infected being able to be seen by the immune system, and so they can evade the immune response. Our HIV program, where we have nice phase 2 data already, has demonstrated proof of concept of our approach. We've had an interest in SARS since the early days of the SARS-1 outbreak. In fact, Biotron scientists were the first to identify the coronavirus E protein as a viruporin and purported as a good target for drug development. During the last 12 months, we've been very focused on further expanding work to extend to SARS-CoV-2. And just to really, without going through the details on this slide, I think it's really important to know that we're targeting this thing with E proteins, protein found on all coronaviruses. And it's especially in SARS-CoV-2, it's really critical because this E protein that's found on this virus sets up and triggers an inflammation cascade in the lungs. So you have a normal lung alveoli. If SARS-CoV-2, this E protein intact is there, you get all of these cytokines, these pro-inflammatory cytokines racing to the site, and that then makes being released to the site. And then you get various cells, other immune cells getting in there, and that causes all of the congestion, the inflammation, and all of the things that then result in severe disease and respiratory distress. We do know that if you delete E protein sequences, you attenuate or reduce the infectivity and the pathogenesis of coronaviruses. So we had expected that by developing our drugs, which are viruporin inhibitors, targeting the E protein, that we would have an impact, would be expected to have an impact on moderate to severe COVID. During the last year, we've been focused on making some new compounds based on our original SARS-1 work. We have some really nice new compounds that have very nice activity. The problem with these is they're actually not yet ready for the clinic. We expect they would be soon. So we decided to also run through BIT225, which is our lead antiviral drug, and check that to be sure, even though it's a different chemical class, it's slightly different than the other SARS drugs we've been developing, to test them in cell-based and in vivo animal studies of SARS-CoV-2 infection. And these studies, I should say, are undertaken at the Scripps Research Institute in La Jolla, in the lab of one of our very good collaborators. The model we use, this will be on the slides, and most people won't be that interested in this, but it's the details of it. But it's a really interesting mouse that's recently been developed that been engineered to express the receptors that SARS-CoV can get into the cells. And you end up developing a dose-dependent lethal infection of SARS in these animals. They get very rapid weight loss. They normally die within about 12 days. They shed high levels of virus from the respiratory tract. They develop pneumonia and lung pathology similar to what is seen in severe cases of COVID. And you get the immune cell infiltrates and the pro-inflammatory cytokines, or the cytokine storm, that's similar to what you see in human COVID. So it's a new model, but it's now being used to study the pathogenesis of the disease and to assess therapeutic agents and their ability to treat COVID. The data that I used today is with our lead drug BIT225. So the study was done at 2 different doses, 100 mg per kilo and 300 mg per kilo. It's given by oral gavage, that means just mixed in methylcellulose and dosed to the mice, compared to methylcellulose without 225. They're infected with the virus intranasally, dosed twice daily without drug for 7 days, and then endpoints looking at body weight, clinical observations, how they are, and measuring the amount of virus reduced or present by 2 different methods, and also looking at the pro-inflammatory, the cytokine storm in the lungs and in the blood. The really important, this is probably one of the most important slides we have. What we found is that BIT225 protects against severe disease in these mice. What this shows is the days post-infection and the percent change in body weight. Ordinarily, these mice, they're juvenile, they're still growing, so ordinarily their weight would increase. If you infect them with SARS-CoV-2, they get very rapid weight loss, and that can stop the study at day 7. But if you continue this out by day 12, they've lost more than 25% of their body weight, and basically it's a lethal infection at that point. And this weight loss is loss of body weight is due to all of the various things that are happening in their body due to the disease. What we found with 225 at 2 different doses, the dotted line shows the lower dose, which is 100 mg per kilo. The higher dose is 300 mg per kilo. Is that these mice were very healthy throughout. They continued to, expand their body weight, is what we expected. And there were healthy mice with no evidence of disease. We can see there's a dose response, there's starting to be a bit more maybe impact on body weight with the lower dose, but certainly the higher dose, these mice were very healthy. I should say as well that we actually, because this is a well-studied characterized drug in terms of the doses that we give in humans, that these 300 mg doses is in the range of the dose that we've been giving in the human studies, other studies we've done for HIV to date. So this is a really significant finding. A lot of the other drugs that have been tested in this model reduce the virus levels, but reducing virus levels doesn't always translate into protecting against development of severe disease. So this is a really significant and clinically relevant finding from our study. The other important one just to highlight is the cytokine storm. I showed the slide with those alveoli in the lungs with the B protein sets off all of the IL-6, IL-1 alpha, beta, TNF alpha, all of these cytokines get stimulated and produced in the lungs. And the black line here in all of them is the control mice, which get the methylcellulose without drug, and the levels of the various cytokines. And the red is 100 mg per kilo, 225, and the blue line is 300. And you can see in all instances, there's a significant reduction in the levels of these cytokines. And also really pleasingly in a dose-dependent manner. You give more drug, you get more of a reduction. And this matters because, as I've said, the increased levels of these cytokines are linked to severe disease and death in people infected with SARS-CoV-2. And controlling this cytokine storm is essential for successful treatment of COVID. This is the same thing, we won't go through it, but it's mirrored in the blood the same way as what you see in the lungs. And the final point is that there was a significant reduction of virus levels in the lung and the blood by two different means, by looking at viral load and infectious titer. And what we saw in the lungs is mirrored in the blood. Once again, on the control mice infected but not treated with drug, 100 mg and 300 mg of 225. So this log reduction is around 3 or 4 log reduction that we've seen in the animals is very good. It's on par with the other drugs, or better than the other drugs that have been in that are in development for this indication. In summary, we have generated substantial and clinically meaningful efficacy in this model, significant inhibition of the virus, reducing the pro-inflammatory cytokines, and reducing the severity of complications indicated by the general health of the animal, preventing weight loss. The really important thing is this is not a drug that is a newly made, still got to go through safety studies, still got to go through manufacturing and all of that. This is a drug that we have a lot of experience with in the clinic. It's currently in development for HIV eradication and improving health outcomes associated with immune dysfunctions. We currently are running 2 phase 2 trials, one in Thailand in ART treatment naive and one in Sydney in ART treatment experienced populations. To date, we've completed 9 trials, over 200 people have been dosed with up to 12 weeks of dosing, current approvals, no existing trials with up to 24 weeks of dosing. And to put that in context, for COVID, if you look at the other drugs that are being developed and the trials that are being done, you're only looking at like 5 days of treatment. So this is COVID and SARS is in many ways a significantly easier proposition in terms of designing and undertaking trials than HIV, which needs to go on for many months. We have a very well characterized safety and PK profile. The drug at the moment is suitable in the existing trials for once a day dosing. We've completed all the needed safety studies, complying with FDA requirements along the way. As luck would have it, during this last 12 months, we've done a 3rd manufacturing campaign with our manufacturing contractor and have on hand around 8 kilos of GMP grade BIT225 drug product on hand. That's over and above what we need for our current HIV trials. So we're in a very good position at present. The other important point is that 225 is a very, very stable drug. In fact, we've had its stability out for 7 years with no degradation, and it's stable at room temperature. So what we are doing at the moment is we're aiming to progress this as quickly as we can into human studies. We're in discussions with international advisors to design clinical studies to assess the efficacy against COVID quickly. And that's likely to be potentially a study ex-US, outside of the US, in patients who are high risk and potentially unvaccinated, though that's still to be determined in conjunction with our advisors and principal investigators. And that can be done very quickly while we can engage with the relevant regulatory authorities, particularly the FDA, through their CTAP, which is their Coronavirus Treatment Acceleration Program, to facilitate fast assessment and progress into a larger study through an IND process. And we are planning to do everything we can to progress this as quickly as possible into studies. I'll leave it on this slide, but if anybody, which just summarizes what we've said, but if anybody has any questions, then Rob and I are certainly happy to address those. So we have some questions here. There's a question here from Colin Sharp. What is the base level of these measures in a healthy animal? How close to normal does the higher dose achieve? Colin, we're still in the process of determining that. I think the main thing is not so much the quantum of that, but the fact that the overall health of the animal indicated that there was no underlying pathology, that they were healthy. There's a direct association with loss of body weight in these animals and development of disease. So I think that at the moment is the really key measurement, and we will get some more information about this information as we go.
It is a placebo-controlled study.
That's right. Richard Keller, and it's nice to see you on this call, Richard. Will we be able to fast track human trials? Yes, that is the aim. In particular in the US, the FDA does have a fast track program, CTAP, as I've mentioned. They are very enthusiastic to have more direct acting antivirals through that process. There's quite a few immunotherapies which are not related to direct acting antiviral activity in any shape or form that they're not so excited about. They want more direct, small molecule direct acting agents. I think, Rob, have you got some comment about that?
Yeah, I mean, the FDA, and it's actually the program is really run by the NIH, and they're behind the game in terms of treatment. They did the remdesivir trial. A lot of controversy about that lately. You may be reading about that. And they really didn't pay much attention to oral therapies and put all their effort, or the bulk of their effort, into vaccines. And they really pushed the vaccines. The vaccines got developed, as you know, very quickly. Well, you know, we got problems with the vaccines. We got variant breakthroughs. The vaccines are not going to be the answer. We don't even know if we can ever reach herd immunity. So there's been a shift back to the oral therapies. And the program that Michelle discussed is now actively trying to recruit small companies that need assistance with design, development, and implementation. They have a network ready to go to test these drugs. So it's very exciting.
Yeah. Nathan Watts has asked a question about funding. And yes, he's noticed our bank balance. So the funds that we have at hand are sufficient to fund our existing HIV trials. That's what that money is earmarked for. We will be seeking non-equity funding through various government agencies in Australia and overseas to fund as much of this as we possibly can. Once we get a better understanding of exactly what we're looking at, exactly what we are looking at in terms of funding requirements, we'll be in a better position. But certainly there, in particular in the US, there's significant non-equity funding, grant money available for this. I think we're making sure that we can access it and get this underway as quickly as possible. And this question here about rough timeline of fast tracking. Look, maybe Rob's got some comments on that. We don't at the moment. This is new. This is really new data for us. We've been working, trying to get an understanding of this, filing patents, preliminary discussions with our US advisors, is putting on a regulatory affairs person to work with us through the FDA programs. In terms of fast tracking and the timeline of that, I don't have an answer for you right now, but I have been told that it is a very fast process and that you get immediate responses. Rob, do you have anything more on that?
Yeah, let me add to that, because you look at the speed that Paxlovid, the Pfizer combination drug, and molnupiravir, how fast they were developed. It really was, it's less than a year, start to finish. Now their programs existed a long time before that. Molnupiravir was originally developed for influenza at Emory, a little bit in hepatitis C, and then they licensed it to Ridgeback Pharmaceuticals in Miami for an influenza drug. And then along came COVID, and it very quickly got a partnership with Merck, and did their study in a matter of months. The Pfizer group kept very quiet about the development, other than saying they were developing it. That program too had started a while back, actually with the original SARS, but they were focusing on the protease inhibitor, which is different. I don't know if that's why their results appear to be better than molnupiravir or not, but they had this program all set up, and that's the first drug, the active antiviral part, that was developed specifically for SARS-CoV-2. And that's only in this calendar year this happened. So if this actually works in humans, it could go very quickly if the funds are available.
Yeah. I think the other thing is if we are also looking as well in parallel with the FDA to move to do a smaller study outside of the US, like we can do that very quickly. We have material on hand, we have potential sites on hand. We're just conferring once we have some more information on that in the public domain, we will. There's also another question here, I think from Adam Kostick, asking how do these results compare with other antivirals at the same stage of development? I think my understanding is, and Rob will correct me if I'm wrong, that when like the Merck and Pfizer drugs were being developed, this model wasn't around. So I don't think it was initially tested in this, in particular the Pfizer one. In terms of what we heard, what is being published and what we've seen and what we've heard from our other US advisors when they've looked at this data, this is as good as anything anyone has seen, particularly because knocking down the virus in large amounts like remdesivir isn't enough. We know that because remdesivir in the clinic has been disappointing. Fabulous as an antiviral in this model in knocking down the virus. No effect on the severity of the disease though in patients, and also impact on the cytokine storm. So the fact that we have seen significant things that relate to potential clinical benefit rather than just knocking down the virus is what really excites us and compared to what else is there. Richard has asked, what are your gut feels of success in human trials? Look, Richard, it's always tricky. You know, I think the thing that we have is that this is a drug, at least that has been in the clinic. It's significantly de-risked in terms of safety, toxicity, all of the other things that can derail new drugs. That's taken care of for us. And we're in a very strong position because of that. Technically, will it work? All we have got to go on at the moment is the data in the mice, which certainly look compelling. It makes the way that the target that we're going after, the E protein, and its central role in the pathology of all of this gives real credence to optimism for success. But we need to do the study. We need to do the study.
Yeah, the pathway for this development is fast and furious, and you're going to know very quickly about the effect in humans. But there is activity in 2 other RNA viruses, HIV and hepatitis C. So I think it's definitely justified to move forward in human trials as quickly as possible. The first trials, we understand the PK, that's all been studied. All the cytokines in HIV have been studied. So we can leapfrog into the development basically at the 1B, 1B2A level, which does not involve a huge number of patients. Typically, those studies start in the 30-person range to see if there's any signal at all. The pathway is pretty clear.
And then after that, we'd be then in parallel with that, going with the FDA, talking with them and setting up on the base, to then do phase 2/phase 3, the way that the Merck study has been done. Once we get some indication in a smaller study first, which is basically the way that the Merck and Pfizer drugs have been developed as well. And Colin has asked a question, does this result have any implications or influences on the utility of the drug on the other viruses? I'm not quite sure if you're talking about from a commercial perspective, perhaps. Like we're very committed to the HIV program, where it's an absolute unmet need. We're very excited by the program, very excited by the potential results. The data we've had to date in the last phase 2 was quite extraordinary in terms of, looks like we're unmasking the virus so the immune system can then attack the virus in those reservoir cells. and improve the health outcome in patients at the same time. We've got those 2 trials to really put the final stamp on that, are in progress. You've seen our announcements with the timeframes for that. We expect to complete the clinical phase by mid next year and have data not long thereafter. We're committed to that. And I think that what this additional work now with COVID as well shows that it really is a platform, that it is a whole approach to treating different viral diseases. Also point out that while 225 is our lead for this, we can move into the clinic very quickly, but it's not our only compound. We have a whole lot of other new drugs as well in development behind this, which are looking really nice. They're getting really nice data as well in these mice. They're getting safety data, preliminary safety data, and various systems that we have to do so we can get a lead to go to the clinic. Well, we were planning that process before 225, but it means we do have other compounds as well there. So and same for the next gen for the HIV. We have a very solid hepatitis B program. And for all of these, our approach is direct acting antivirals that knock down virus levels and unmask the virus and boost the body's immune system by impacting on immunomodulation. And I think that this story is really coming together very well. Yeah. So I think unless there's any other questions, I think that that might be it, actually. So thank you very much to everybody for attending today. Appreciate your interest, and we look forward to bringing you further developments in the near future. Thank you.